Do we have a first group? I mean, we've got slide shared. Is there anyone here in person who's part of it? Or online? I think you're muted, whoever's sharing.
Sorry. Oh, okay. Sorry for the delay. Okay, thank you. And hello, everyone. I'm Nuye, and my group project is about the NSD imagery data. And I got a cool title here, like the what survives in the mind's eye. And for this project, this process is trying to ask a very simple question, like when the same person first sees an image and later imagine it, which parts of the brain representation remain organized in the same way? And this is actually a very difficult question, and we didn't manage to give a very satisfactory answer, but we tried to approach it from two sides. The one is from RSA, the representational similarity analysis. Another one is brain decoding. All right. So let me give a bit introduction of this data set. The first is, so in this data set, we have 8 participants. And for each participant, they undergo several trials. So before their fMRI scanning, each participant learned 18 targets, and also along with their cues. So the cues look like this. So the first 6 are just simple shapes, like bars, rotated and rotated crosses. The 7th to the 12th are the natural things or some artworks. There are actually 6 other cues, but I've not displayed them here because that's a bit irrelevant to this vision imagery task. All right, so here for each so in this data set, there are actually like 12 rounds. So for each round, there in total, there are around 576 behavioral trials, which are participants seeing an image or imagine it with their brain responses measured. So for each measures, the experiment record the betas, which measure the amplitude of the brain responses. So there are in total 720. So for among them, only like 400 are related to the vision versus imagery task. So we select this out. Okay, so overall, this is kind of a small data set. So in principle, we're not able to like train a very fancy model on it. So basically, the most straightforward things we can do is try to learn something from the bigger data sets, like the original NASD, and let's see whether that result transferred to the NSD imagery. So let me pause for a little bit, because in case if you haven't seen the original paper, I would say the original paper is much cooler than my work. In the original paper, they try to first learn a fancy model mapping the brain responses to the actual images, and see that whether that model can recover the signal in imagery, and see what picture can be recovered from your brain signal when you're just imagining it. So see here, we can see that when people just imagine surfing, the recovered images are also about some person doing something. So that's not closely related, but somehow they share a course-related common semantic information. So like the original paper tries to use this computer vision model to decode your imagery brain signals, which are pretty cool. All right, let's get back to this work. So for this work, I try to test the information transfer 3 levels. The first one is the same region RSA. We try to see the representational dissimilarity matrix during vision and during imagery, and see whether these two are correlated. And for the second part, we'll do some cross-region RSA. See like whether the visual cortex information transferred to the parietal cortex. The third part is about brain coding. So for RSA, so here I calculate the beta average across subjects and voxels for each subject, and then do this are RDMs, the representational similarity matrices between different targets. So as in each set, remember that set A are for simple shapes. There are 6 different shapes. Set B are for natural things. And there are also 6 different things. So these are 6 by 6 RDMs. And we want to see whether the RDMs from the actual perception is similar to the RDM to that in imagery. We especially, in particular, we do this among different regions. So here, the NDS general are kind of a combination of the early visual cortex and the higher visual cortex. We see that for simpler shapes, the signals aligns more well. So the highest row, the correlation appears in the early visual cortex, and which is about over 0.4. However, for set B, which are natural things, we have more richer semantic information. We say that the highest row correlation appears at the higher visual cortex. So this is kind of compatible with our knowledge in this difference between the two cortex. That's the lower shape and transfer more through like the early visual cortex, while the more complicated patterns may transfer beyond that. But however, I need to put a caveat here. For this small data set, the statistical evidence is quite low. So that's why also the author is calling for more data that maybe help us to analyze this better. We also tried the cross-region RSA. The basic conclusion is that we see that if we only look at the visual region to parietal cortex transfer, there are some information loss, but the loss is not statistically significant. So let's say for this data set, if we only look at the visual, we look at the visual to vision to visual RDMs, they look quite similar to the vision to parietal RDMs. So that may indicate that these two kind of align. All right, the third part is about the brain encoding model, where we try to predict the brain responses with some models. In this part, I use some, the result is that we fitted on the original NSD dataset and try to use that fitted model to predict the brain responses during imagery and see whether this model still predicts well. We see that if people actually see the images that's during the vision stage, the R-square are much higher. While comparing to that, if you just imagine it, the prediction performance dropped much significantly. But overall, the two performance are still relatively low, because I guess that we still have to improve the model to make better prediction or to collect more data. So if you're interested for some works that really do the encoding model well and do the prediction well, I got some good papers. All right, so that's basically just 3 parts of my work. Given that although we don't have much group member here, but I need to put out this special acknowledgement section to thanks Tamar, Tongla, and someone who prefer to be anonymous, because although they did not contribute to this code base, they provide very much insightful ideas for this project. So thank you. And also this data set, I have put this data set in the repo. So if you want to try to download this data set, my repo help you to build the processing pipeline. So you can, if you're interested, you can simply try this. All right, so I think that's all for my project. So thank you.
Amazing, thank you.
All right, self-supervised learning is up next. And precision fMRI, your deck following that.
Is it? See the screen. I lost it. So. Sure. I'm not sure where is it. Oh, this one. Okay. Okay. Hello everyone. We are a group of, my name is Junbom and I worked with Federico on this project. Our project is on self-supervised deep learning for structure to function prediction in visual cortex. Yeah. Only visual cortex has a strong coupling between visual field position and brain anatomy. To be specific, neurons in the visual cortex respond to specific location in our visual field. For example, this notch on the Kirkland circus responds to 15 degrees of eccentricity very consistently across subjects. It is very useful, but we need an hour of fMRI to get this kind of information. So, to overcome this, templates have been used to predict retinopathy from brain anatomy without functional MRI. So, these templates defined in standard space has been projected into individual spaces to get individualized templates. However, the biggest limitation of this approach is that it is highly homogeneous across subjects. So recently, geometric deep learning models have been used to predict retinopathy from anatomy using the curvature and binding features on surface and its connectivity information on a mesh. They use geometric deep learning models and predicted 3 kinds of retinopathic maps that is also defined on a mesh. It has been reported that geometric deep learning improves prediction of retinopathic maps, and I replicated this result previously. So in these maps for the polar angle, there is some unique patterns in individuals, and template seem to interpolate those reasons following the template. On the other hand, geometric deep learning predicted these subtle individual patterns more effectively. Correlation was also higher for the geometric deep learning. However, there are still limitations. Oops. Yeah. Yeah. However, there are still limitations. First, deep learning requires a lot of retinopathic maps, but we still have a few label samples. We try to recruit as much data as possible, but it was around 500 samples. A label data scarcity also appears in other domains, so these domains try to solve this using self-supervised learning approach. Yeah. Oops.
So folks on Zoom, we're having some technical issue where our screen is blinking out, kind of like it did in Surge's talk. Okay, let's see if this.
So the idea of self-supervised learning is that using the unlabeled data, which can be used as an input for the supervised learning, we can learn more fundamental information from this unlabeled data. So in this project, we tested the feasibility of self-supervised learning to improve retinopathic mapping modeling by doing this learning for structured data. From now, he will continue.
Thank you. Specifically, in this case, the key observation to understand self-supervised learning is that we have a lot of unlabeled structural data. So good quality data for which we don't have a retinotopic map. So the idea is to use them to learn general structure of the surface that basically contains some sort of general properties that are not related to vision. Okay. And then this happens during the pre-training step. So during the pre-training step, we really aim for a general representation of the surface that is later fine-tuned by incorporating vision-specific properties from labeled data during the fine-tuning step. For prototyping reason, we have reduced pre-training to the unlabeled subset of ACP, which is around 900 subjects, and fine-tuning to the labeled subset of ACP, which is around 161. This is not ideal because self-supervised learning really works when you have a lot of unlabeled data, and evaluation should really be done on data that come from a different distribution, but still, this was the best we could do in 4 days. In this slide, you can see our self-supervised pre-training pipeline. The idea is that we start from a mesh that represents the surface derived with free surface from the ACP data. We project it onto a sphere, and this is done mainly to make the next step easier. We split this sphere into triangular patches, and we mask 60% of them, meaning that we replace them with a learnable token. And the transformer model is trained to reconstruct the missing patches from all the others. The idea is, again, that this way you are able to capture the general properties of the surface and still exploit the unlabeled data, even if you don't have a topic map from that. This model is called the SIT. It's an adaptation of the vision transformer to surface data done by Daniel Ruckert and Emma Robinson. And this is our fine-tuning pipeline. It really starts on the very same data. The main difference is that we are doing no masking in this case. So we feed all the patches to the model, actually the model that resulted from the pre-training step. So it's not starting from scratch. It can leverage that sort of general representation that was extracted during pre-training. And basically the next step serves this purpose of injecting vision-specific knowledge into the representation, coming up with a single scalar for each vertex that represents our prediction of polar angle. In between, there are some very technical steps that basically serve the purpose of going from a patch granularity to a vertex granularity, but the idea is still that in the end, you come up with a single scalar for each vertex and you use it to compute an L1 loss to train the model. So what kind of results do we hope to get? Well, the key idea of self-supervised learning is really to give access to the model to additional knowledge that is present in unlabeled data that we could not otherwise exploit with a supervised approach. So when we start the fine-tuning phase, when we train on the labeled data, the model has to learn less details of the surface representation because it has already been exposed to surface data during the training phase. And so overall, this should mean that we can get the same performance on the retinotopic map prediction with less epochs. And this slide shows that it's kind of working. On the y-axis, we have the circular correlation between the prediction of the model and the actual polar angle values. On the x-axis, we have the number of fine-tuning epochs. And as you can see, the red curve that represents the behavior of the pre-trained model is able to reach, and here we have a zoomed version, is able to reach the same performance level with less epoch compared to the orange curve that is the model trained from scratch. And the interesting thing here is that it looks like we are also able to get slightly better performance than the state-of-the-art model, which is deep retinotopy. Don't take for this, no. Yeah, in general, these results were obtained on a single seed, but we have replicated them on multiple seeds. So we have done a sort of cross-validation. There is actually a bug in the computation of the left hemisphere representation, but this should not affect the overall trends and the ranking between the different strategies. It could affect the maximum value of correlation that we are able to get, but only for the better. So what do we get out of this? We have a fully functioning pipeline for self-supervised learning on mesh data, which is a good starting point because self-supervised is usually applied on 3D volume data or 2D images, so very different data. It looks like it's working, but obviously to reach a statistically sound conclusion, we have to train on many more data and a much more heterogeneous data set. And finally, thank you everyone for these 2 weeks. It has been a marvelous experience.
Thank you so much. Folks on Zoom are having a little bit of technical troubles, but hopefully they're over. We'll see. Next group. All right.
Just a disclaimer, I have a cold, so I'm looking really nasally and congested. I'm sorry. But hi, everyone. So our group worked on precision network mapping and multivariate analysis in the PAM dataset. So I know some of us are familiar with that dataset, but just a quick little reminder that the precision targeting of association networks dataset is a large-scale precision fMRI dataset from the Graton lab that uses individualized functional network mapping to study variation in human brain organization, and cognition. And our group specifically was interested in memory. So just a quick outline, what we did for this past week was individual network replication, a little mini side quest that you hear about, and then we also conducted some GOMs and multivariate analysis.
So I'll first talk about the individualized network definition. We as a group are interested in the default mode network that is very relevant for episodic projection and memory. One interesting thing about the default mode network is that the constitution, the structure, the functional structure of it varies across people. And on the left panel, the purple regions are a canonical definition of DMNA. And on the right-hand side is a data set called HTTP. It shows across more than 300 participants, the variance of network functional structures across participants. So a brighter color would mean there's more variance across participants. And the essence of the figure is that some regions have more variance, some regions have less variance, and DMN has the highest variance. So we would like to see if using an individualized definition of DMN, would that give us a better analysis results? And how to define individualized network? It needs to go through a very complex pipeline. First, surface reconstruction, functional connectivity matrix, designing individual networks. We hit like 5 different walls in that. And as we are bending our head to the walls, this angel on earth came to us. It's Katerina Gretchen, the curator of the Penn dataset. She very generously shared with us her definition of individualized network results. So we came to here. On the left-hand side is the canonical definition of DMNA. On the right-hand side is the definition of individualized DMNA across 6 different participants. Just visually scanning through it, we can see that there is variance. And our big question is, who will win, the canonical or the individualized? And we'll pass it to Kari.
Hello. So, for the task fMRI, we analyze the data from the episodic projection task. And in this task, participants are given 30 trials, and each trial is a scenario that's either an event about oneself or another, either in the past, present, or future. And in our study, we were interested in those about the self, distinguishing specifically future from past self, which would recruit the anterior default mode network A for future self versus default mode network B for the past self. So, next slide, please. So using the task GLM, generalized linear model, rather than averaging all the trials in a certain category, we modeled brain activation, whole brain activation for each trial on a trial by trial basis. So we could keep the beta weights for each trial and use the most amount of information in the model. And in this case, after we get the beta weights for each trial, we transform those to server space. And basically, this is what we're feeding the model is a beta weight for each, we give you a brain map with a beta weight, with a beta map, and see, is this person thinking about the future or thinking about the past? So there's a past self or future self. All right, go on. So the model itself was ran with a multivariate pattern analysis, and cross-validation was done with a leave one out method to predict either past, present, or future self from these trials. The task data was masked between either the default mode A or the default network B using the 2 different methods of defining networks. One is the individualized network masks, and the other is from the YAO network masks. And we see, regardless of which distinction, which atlas, YAO or the individualized network mask, there really wasn't any difference in accuracy. And there really wasn't, the default mode network B demonstrated greater within subject variability as opposed to the individual masks. So now I'll hand it back over.
Oh, there. Okay, so this project was my first time working with the fMRI data, so very interesting. But also, I was allowed to be a part of a group, but also go on a tangent on my own. So I did that. So my first idea was that I was interested in, since it's precision fMRI, I was interested in just when we accumulate data, how does that affect reliability? So my first plot is just taking the resting state fMRI data and splitting it in two halves, and then how to put it, get a target from one of the sets and use the other one to measure like how does reliability evolve as you add more data as you go. So the dark red line here is just showing that like the same individual as we add more data versus the full other split, and the other one is compared to the nearest other subject. So how to put it? I guess the the takeaway from this is that brains are quite similar between the nearest other subject and the individual we're interested in. But there's also a gap between them. So there is, of course, some individual signal as well. Then I moved on to like, I guess we're interested in brains, but we're also interested in, as I said, the individual. So what I did was take first it's the same line as the top line is the same as the other plot, but then I also took just average of the 9 other subjects and subtracted that. So basically just to try and see like the individuality, or like what's left of the individual when we subtract the whole average brain from the other subjects. And I guess lastly is a different view of the same process, which is upon or subject number 1, which was the subject with the most rest data. The top plot here is the whole map, where we start with 1 minute of scan time, 5 minutes, 20 minutes, 45 minutes, and then 90 minutes, and just seeing how reliability evolves as we add more data, or the covariance matrix evolves. And then this is the bottom one is the same as I just explained. It's removing the average brain from using the 9 other brains. And I guess it was very interesting just seeing how there's still movement going on, especially between like 45 minutes and 90 minutes, although it's not heavy patterns, but there is something going on. And especially since we're with precision fMRI, we're interested in the individual networks or the individual's networks. So I think that's, I don't know, I think that's what I have.
All right, so as Tonga mentioned, we did run into some hiccups and challenges. We had to ask Katarina for help, which she graciously offered. But some challenges that we went through, even though there were some challenges, there were also takeaways. So what we learned last week with GitHub and working with OpenNeuro and even NiLearn and everything else, it was like a really interesting experience. Like we had like a couple brains together, but it took us a while, but we managed to get through. I think it was a great learning experience to use these resources in the future for our own work.
And just in conclusion, we developed a pipeline for MVP analysis within individually defined networks using the PEN dataset. We found no significant difference between individualized versus canonical networks, although with the limited sample size we have, it's really hard to make conclusions and interpret too much into it. So thank you so much.
Amazing, thank you. I believe up next is the quality assurance websites. And temporal discounting, you're on deck.
I guess we're the quality assurance website. So it was myself, Jan, Jenny, Hendrik, Tamaya, and Mira worked on this for a week. So very briefly, can anyone tell me how your lab currently teaches you quality control? Anyone? Generally, I'm hearing we don't. Very cool. So, pretty much every lab reinvents QA training from scratch. So there's no good standard way to teach artifact recognition. EEG and MRI QA are in completely different silos. And there's a lot of good material out there. It exists, but it's pretty scattered. Sometimes it can be really hard to find, and it can be pretty inconsistent. I know I've gone through some data and found 50 different opinions on how to reject artifacts, and I'm sure you've done the same. And importantly, a lot of new trainees learn by trial and error and not by any like structured design. So our goal here was to create an open educational resource for neuroimaging quality assurance. So we wanted to include things like annotated artifact examples, visual inspection guidelines, and some curated tools for both MRI and EEG. So our first issue is that we could not decide on a name for our website domain. So if anyone would like to scan our QR code and come up with a suggestion, Henrik will be absolutely furious if you started with Neuro. So what we wanted to include were for both MRI and EEG, we wanted to have some links to different resources that include some like base foundational articles and also packages and tools that you can guide your QA pipelines with. Some descriptions and visuals of common artifacts that you might find in your research and how to identify them and address them. Some different QC workflows or checklists that you can apply to your experiments, and some practice questions to test yourself. So I am gonna pass it to Tamaya. We are gonna do a little walkthrough of the website.
I believe we send the website link to the Slack channel, so feel free to click on it and explore by yourself. And
Yes.
Tamaya, are you ready?
Yes. So first in this walkthrough of the website, we'll be going over the EEG open resources here on the screen. And first, we organize the resources into 3 broad groups here. So we have methods, artifact detection and rejection, and packages and tool. First, methods kind of focuses on a little bit of the articles on choosing and evaluating EEG methods, because we know QA and pre-processing can look so different across many studies. And if we go back and go into artifact detection and rejection, we'll have resources here on recognizing, rejecting, and correcting EEG artifacts. And I want to also kind of show the packaging, just briefly, packaging and tools. And here we have links and also suggestions in the blue box of how to use these tools as well. And then we have the websites, documentations, and also instructions on how to install some of them as well. So if we go back to artifact detection and rejection, for sure. I just wanted to say, like, we have our recommendations, but also that if you need a resource for a specific artifact, we may have it here and you can use the search box that we've added. But right now, we only have, a lot of these are for ICAs, but we're really hoping we get more resources, but really provide an overview and a synopsis of each article, what the resource is, what is most useful for folks, and if the PDF is available, we've linked that as well. And so methods and packages follow the same layout, but we're still working on adding and finding more of these resources, and welcome any suggestions as well. So now I'll pass it over to discuss our artifacts page.
Thank you. Hi, everyone. My name is Chen Ye. I will briefly introduce the 2 key components in the EEG section. So you can see in the website, we have artifacts and QC check. So in the artifacts, we developed 5 different categories of the common EEG artifacts. So we provide not only just like the figure of the eye blink or one single EEG artifacts, we also provide like what you will see when you see the raw data, how will the eye blink look like. And also we have judgment calls and decision-making considerations. So you can ask these 4 questions when you see your raw data to make a judgment whether this is eye blink or just like regular EEG waveform. And we also provide some possible solution and responses, because every lab use different equipment. You may have different analysis technique, and also it depends on the PI preference, because some PI don't like the ICA, some PI prefer more restricted analysis. And so except the ocular module, we have eye blink, vertical eye movement, horizontal eye movements, and we have the muscular part. We provide two common artifacts in EEG, and we also have more artifacts like the electro-bridging and noisy or unstable channels. We want to provide not only for the data analysis, we also provide some suggestions before you do the data collection. And we have another very important key component in the website. It's called QC checks. So we already complete two modules here. The first is quality check workflow. And we want, when you click these modules, you will see we provide a central role and before you do the workflow. And we have different components, the different steps you can follow and to do the analysis in the raw data. And we provide for quality control stage, so you can check and make decisions on your raw data. Okay, last thing I want to emphasize is we have the QE checklist. You can just click and download in your computer. When you open, you will see like the documents you can print and send out to your RA for helping you to do the data collection. Okay, I will pass it to the Ken.
All right. Okay, so Meryl and I work on the MRA page. So the format basically follows the EEG section. We have open resources, artifacts, QC checks, and also a small practice section. So it's basically the same format as the EEG section. So put together some resources, and it includes papers, software packages, as well as tutorials. So feel free to explore. There will be more added to the section. And this artifacts section is also work under construction. But here's a brief overview. I will say the biggest challenge in compiling this list of artifacts is the licensing of those images, because Mackenzie has a very useful paper on artifacts, but the publisher has restricted the reuse of and reproduction of the figures. So I had to find those open access figures or produce my own figures. So these are figures I screenshotted from my own data set. So I had good brains and bad brains, as well as some judgment call decision making checklists. These are all from my own data set. So here's also a decision making little guideline and citations. It's very similar for other artifacts, and more artifacts will be added to this page. And the QC checklist, it's also similar to the EEG page, but I put everything into the website format so that it's easier to just check and uncheck it. Yeah, and for the practice, there's a little quiz that you can practice doing QAs. Yeah, I think that's just a brief walkthrough of our website, and everything is still under construction. So we have some final slides.
Yeah, we're gonna go super quick. So we want to add more modalities to this. If you do MEG or like PET scans, anything like that, we would love it. We would like your submissions. If you have any feedback, please send it our way. We would love to make this a resource that's really useful to everyone. We'd love to include like a teaching data set, because it was really hard to find a lot of this data like open source that people can practice on. And we would like to make it a little more comprehensive for everyone. Thank you to all these people who shared their resources with us. You could be on here if you shared your resources. We would greatly appreciate it. Thank you.
Thank you. Next up, we have how to predict temporal discounting, and on deck we have whole brain dynamical modeling of character orientation. Yeah. Sorry.
Yeah, so our project was titled How Not to Predict Temporal Discounting. Our project was more focused on as an exercise, a learning exercise. So don't expect like high performances or something like that. So the actual question we had was if we were able to predict temporal discounting using cortical functional productivity during a gambling task, as well as white matter diffusion methods. We had the data coming from the Human Python project, having access only to the open access data. And the actual target was the undervaluation of the rewards over the delayed period of time. So coming from an amount of 40K, this measure allows us to see how this value decreases in subjective value across time. So 40K now, it's equal to maybe 20K at 10 years. So it's been seen in other publications that people that have like this steep decrease in value, so in 6 years or 5 years, for example, this value decreases a lot. It's the same people that have like more impulsive tendencies and low self-regulation. So we took this as a measure of the capabilities of a subject as in self-regulation subject. And then the summary of all that is the area under the curve. So this is a single data point that we have for each participant, signaling at this self-regulation capabilities. So, well.
I'm changing here.
Yeah, you can go for it.
Okay. I can just start talking and maybe you guys will see the figure. Okay, so that Danielle just described what we were trying to predict, and I'm going to tell you one of the things that we used to predict it, which was fMRI data collected during a gambling task. And during this gambling task, participants were over and over again presented basically with a card deck, and they saw a question mark, and they were going to guess whether the next card or the card next to it was actually high or low. So if you go to the next one, and they did this both for sometimes they did some blocks where maybe 60% of the cards were actually high. And then other blocks did it where 60% were or more were actually low. And this is supposed to make it so that like you're getting some sort of measure from the participants' behavior about how risky they are, and they receive payouts depending on their choices. Okay, so now that you know the variables we're working with, how do we actually work with the fMRI data? Who we worked with? So we took the subjects, we split them into a training and test set, and we first had to validate that all of the participants we actually used for our modeling had both diffusion data and the task fMRI data from this particular task. So that was the first set of screening. And then what did we use? We used Glasser because HCP kind of made it, so we had to. Okay, and then one of the most important things that we did as a team, as the functional team, was to actually figure out how to work end to end from the raw data all the way to our model. So a lot of it was this learning curve of figuring out how to use HCP and NiLearn. So we were really lucky to have Elizabeth around to help us with that. But we figured out how to create a mesh and a masker, and to fit that masker, how to work with SIFT files to actually extract the time series that we needed, and then to transform them with the fitted masker so that we could then compute correlation matrices that we could feed to our model.
Okay, and we used the, so in the end, we had tons of data points, but only few subjects compared to this. We decided to model elastic net. And so we had multiple levels actually. So we had the training data set and the test data set, but we used first, obviously only the training data set. We've split it into 5 folds. And within these 5 folds, we tested iterated through different hyperparameters. And Then we had like 5 estimations of these hyperparameters, and we used those then on to fit it or to fit a final model that we then used to on the test data set to get like the final idea of how good our model performs. Yeah. Okay. We want to show the results basically, but so I can tell you it doesn't perform very good. R squared is 0.04 something. It's basically using mainly ridge regression because the L1 ratio is near to 0. Yeah. Out of the we had 64,000 different connection measures, and only then you can go on, I guess, because I told this already. Yeah, out of the 64,622 data points we had, 171 were non-zero, and we've pictured the top 20, I think. Yeah.
Okay, and obviously, whether or not you believe how valid our very noisy and small results are, we did see that if you actually look at what the regions are in these top 20 significant correlations, that many of them are related to working memory, which is not surprising. And then we see OFC up there, which was our original hypothesis. So we counted that as a very small, maybe not meaningful win.
Yeah, and what you can see here is that our model is very good at predicting the mean. Come on, man.
Yeah, yeah. So moving on to diffusion data, we knew that from the beginning that structural data doesn't really predict the behavior measures, but we still wanted to have some experience with the diffusion data, so that's why we use this. And we took a similar approach as the functional data, but we tried using like 4 different diffusion measures from the 24 tracks. And we tried different algorithms such as bridge, lasso, and elastic nets. And we did, because our model wasn't working well, we did a lot of validation. So we trained the model with the age to predict age. And we ran permutation tests to see that it is significant, if this is significant from the null. And we also test our model and also ran bootstrap to see if there's any significant feature weights.
Yeah. So yeah, we also messed around with dimensionality reduction. So we ended up working with 25 PCs explaining 90% of the variance. And yeah, this is one of an example of the models we tried. So this one is bridge. All of our R-square were pretty much the same, a bit negative. Randomly, the test R-square was better. So yeah, this was cool. Yeah, so just predicting the mean again. Yeah, again, using 5-fold regression. Yeah. We started working with bootstrapping to see if some of the features might still capture some signal, even though the whole model is not working well. And as you can see, there's not really any features yet working well. And as an example, you see how the PC worked to divide the components. So that's AWS for left incident, for example, for first loading of PCA.
In reflection, we had a lot of limitations, such as lack of access to really specific data types, like age would have been nice, limited task fMRI for this data set, and we didn't use the cortical data due to memory issues and lack of experience. Other limitations we faced. Next one. We learned a lot though. We learned how to access HTTP data, basics of delay discounting tasks and everything. Got really good at pushing and merging on GitHub with our large team, and
that diffusion-weighted imaging is not great at predicting behavior on its own. Good to know, and that 120,000 parameters is just too much. So let's go forward. Thank you to people who helped us a lot.
Say thank you. Okay.
Will the model weights be open source?
Whole brain dynamical modeling is next, but before we start this, I'm going to take just 3 seconds to try to change the resolution on my laptop to see if it stops the issue we're having with the screen.
Can you share the screen? So since we have limited time, I'm gonna start talking while this is getting set up.
We'll give it 2 minutes.
Oh,
But,
okay.
I just want to. I want to see if we can.
I mean, I can still do the general introduction.
Okay, yeah, go ahead.
So our project was titled Dynamical Models of Psilocybin Beta.
Sorry.
So the general concept behind our project is that functional connectivity provides a static view of brain organization, but as we know, the brain changes over time. And so this motivates different dynamic approaches to identify recurring brain states or snapshots of the brain, and so that we can identify how these change over time and how they relate to each other. Oh yeah, we've had it. And so in order to do that, we actually compared 3 different methods instead of just doing one. So we are going to look at co-activation patterns, hidden Markov models, and attractor landscapes. So just to briefly introduce our data set, since it's not open source, it was from Daniel's lab. Psilocybin is a psychoactive substance that that acts on serotonin and dopamine receptors in the brain and is kind of a hot topic because of a lot of potential therapeutic benefits. And so for our paradigm, there were 9 participants, and each of them did 3 psilocybin sessions. And so for each session, they took 25 milligrams of psilocybin, and we collected functional MRI data for the baseline before they took the drug, and then subsequently over 3 points in time after taking the drug.
Okay, so co-activation is essentially one of many dynamic functional connectivity techniques. It essentially just uses k-means clustering to essentially group together like spatially similar time points. And for methodology, essentially we use the 456 node parcellation. It's called the fourth parcellation. It's just scaper, the 400 node scaper parcellation plus the cerebellum and the subcortical for 2 different parcellations. The data was Z-scored and it did have the global signal regressed out. So essentially the method identified like 7 optimal caps that were identified, or basically the optimal caps were identified using the elbow criterion, which is just the point where the within sum of squares stops decreasing exponentially. And afterwards, I essentially just computed occupancy, which is just the proportion of like time spent in a particular cap. And then dwell time is kind of similar. It's just the average amount of time spent in a cap before transitioning to another cap. So this is essentially what those like 7 states look like. You can see some anti-correlated patterns, like for instance, cap 1 and then cap 2. Since these are just cluster centroids, you can just project them back onto a surface plot. And another thing you can do is you can take the atlas and then binarize it, has like a 1D version, and then take like a cosine similarity to essentially start like labeling your caps essentially. And on the right hand or your left, I guess, essentially that's just showing the dwell time and the occupancy, across the drug session from like baseline versus drug. So you can see some differences in like cap 3 to 4 and 5 for both of them.
Thank you. The second method we used was a hidden Markov model. And so this is, I first wanted to compare to see if there was differences in how the caps were derived versus using k-means clustering versus the kind of estimated method. And so I used the same exact method that we just showed, same error criterion, occupancy dwell time. And from there, we kind of compared to see if these were extremely similar. So on the left are two of my caps, and on the right are comparison of a cap previously derived and a cap created by the HMN. And you can see that they are similar, but they're a little bit different. So both of them show that DMN have been activated, but they are a little bit different. And you can see that generally there's differences in correlation across the conservation patterns. The really cool thing about hidden Markov models is that from cultivation patterns and also from raw time series, you can then at every single TR calculate a state of functional connectivity. And so across the entire time series, you can see, it's kind of small, sorry. On the bottom triangle for both of these is the 2 hours of baseline functional connectivity. And on the top triangle, on the top is state 1, and at the bottom is state 6. And so you can see that they're very similar, right? It's like very consistent, but there are small differences. And so you might think, well, this is supposed to be a dynamical thing. Why is this like static? You're showing static measures. Well, something we can compute is in real time how the brain transitions from one state to another and the probability of transition between these states. And so what you're seeing is on the top, functional connectivity across, on the left, you have baseline session, on the right, you have drug session. And you can see as they move in time, you're transitioning between these functional connectivity patterns. And you can see that the functional connectivity is very stable. The caps are not so stable. They're transitioning quite a lot. And you can see various differences in activity. And then we can also compute using graph theory transition metrics. So what is the probability of transitioning between this state to this state on baseline and drug? And you can see that they're very different. From baseline to drug on the very bottom, how they change and switch states are very different from one another. And from this, I haven't done this, but we will then encode and then decode behavioral metrics. And so you can say, does transitioning from this state to this state to this state say something about the person's subjective experience in real time? And we can do this at every single TR.
So this is like a network viewer, which is the same as Daniel mentioned. So this is basically a web-based GL model, which is like a live virtualization. So here we have a Gaussian HMM model, which is like a user's latent state, and the PCA on the data for the one subject data. So on the left, you can see it has a interface where you can select the baseline and the visits across the visits. And you can also see the network analysis, the signal analysis on the bottom over there. And you can also select a node, specific node, whatever you want to see, like how it will work. So we are like trying to improve it, making like a more interactive, maybe later at the later stage. Yeah. And one more thing, these 7 networks are connected with basically, we have 456 parcels, which I forgot to mention. They are basically divided into 7 networks. So as you can see over there.
Yeah, so moving into energy or attractor landscapes, the general purpose of this is to model stable activity patterns or attractors in the energy landscape, governing transitions between them. And so we use an Ising model, which is a pairwise maximum entropy model, to look at these. And interestingly, this is just one example of baseline versus drug, but we saw pretty striking differences between the topology organizations and specifically connected components and how they were organized, but also in the size of the basins. So basically how stable different states are. So we saw significantly flattened landscapes for on drug, as opposed to baseline, which were pretty evenly distributed and much deeper. And so we also modeled, we created representations of this using transition graphs. And we collected a lot of data on the actual graph metrics and saw pretty interesting differences, but here's just, a general visualization of how that looks with each of the different scans. So I know we don't have time for questions, but we do have time to answer question. Okay, we have time for one question if there is one.
This may be the first time in NeuroHackademy history.
Say that you guys won.
Thank you. Any questions? Brian, anything?
Yeah. Talk a little bit about how you go about interpreting what a brain state.
Yeah, I guess it depends for each different method. It's going to depend a little bit. For the hidden Markov model, it's a pattern of whole brain activity, and so it could be functional connectivity, it could be coactivation. These are two different types of states, and they also can have different numbers of coactivity states versus functional connectivity states. And so the state that you use is representative of some type of pattern that you're deriving from the data.
And the difference between the two primary sets of methods are that coactivation patterns and hidden Markov models use temporally related information, but the states that are defined in for the energy landscapes are actually more static states. It's just looking at dynamics.